morning, everyone. I'm Lovelyn Bernardo. Today, part about microbodies, peroxisome, and glysosome. Objective: at the end of the lesson, we can determine historical, microbodies structure and types, peroxisome, function of peroxisome, biogenesis, and lastly, the glysosome function. First, we can discuss about historical. Since the mid-1950s, electron microscopists have observed small structures bodies in cells on morphological grounds have been aptly termed microbodies. So, Insidiadov and Peabody in 1966 coined that the term peroxisome for the microbodies of mammalian system and studies their structure of function. The left peroxisome were first isolated from spinach left homogenate. The example is the mesosphere cells by the Tolbert's group. Glyoxylate cycle containing peroxisome. It's called Glyoxysomes. Were discovered 1969 by beavers and the endoplasm cells of germinating castor bean rhizomes. So let's discuss about microbodies, structure, and type. So what it means by microbodies? Microbodies are spherical or oblate in form. They are bounded by a single membrane and have an interior or matrix which is amorphous or granular. And also, microbodies are most easily distinguished from other cells organelles by their content of catalyst enzyme. So what is catalyst enzyme? It is visualized, which is the electron microscope, when the cells are treated with the stain DAB or 3,3-diaminobenzene. A microbody or cytosome is a type of organelle that is found in the cells of plant, protozoa, and animals. This is the example of microbody structures. The cytosol is also known as cytosomes. The technique of isolation of microbodies from animal and plant tissues includes the following steps. First is tissue are ground very carefully to save microbodies from disruption. Second is the homogenate is treated with differential configuration to obtain a fraction of the cell's homogenate which is rich in microbodies. The enriched fraction is subjected to isopycnic ultracentrifugation on this continuous or continuous sucrose density gradient. Structure: a microbody is usually a vesicle with a spherical shape ranging from 0.2 to 1.5 micrometers in diameter. Microbodies are found in the cytoplasm of a cell, but they are only visible with the use of an EM. In biochemical studies have distinguished two types of microbodies, which is peroxisome and glyoxysomes. So the two organelles differ both in their enzyme complement and in the type of tissue in which they are found and peroxisome. So the peroxisome are found in animal cells and the leaves of higher plants. They contain catalysis and oxidases. Example is diamino oxidase and urate oxidase. In both, they participate in the oxidation of substrates and producing hydrogen peroxide, which is subsequently destroyed by catalyst activity. So this is the example of catalyst activity, the reduced substrate and oxidized substrate. So the glysosome, it occurs only in plant cells and are particularly abundant in germinating seeds which store fat as a reserve food material. and they contains enzymes of glyoxylate cycle besides the catalase and oxidases. Apart from peroxisomes and glyoxysomes, a number of other terms have been used to describe microbodies. It is include the cytosomes, paragmosomes, and crystal-containing bodies. Peroxisomes. It occurs in many animal cells in a wide range of plants. The history of peroxisome, they are first observed by electron microscopy in animal cells 1950s, then the plant cells in 1960. According to Christian Ditoff, 1965, isolated from liver cells by configuration, called them peroxisome because they generate and destroy H2O2. This is the peroxisome. It is a single membrane. The number and size of peroxisomes vary depending on environment. Peroxisomes are 0.5 micrometers in diameter and surrounded by membrane. The highest concentration of peroxisome is in liver and kidney. Proteins are directed to the peroxisomes by a unique signal sequence with the help of protein chaperones and peroxins. Peroxisomes are vesicles that contain enzymes to oxide organic substances like fats. In other words, they use oxygens to break down molecules. The enzyme in these organelles produce hydrogen peroxide. Hydrogen peroxide is broken down right away by the enzyme catalase into oxygen and water. It is the location. Inside the cell, which is we can see the plasma membrane, peroxisome, cytoplasm. Peroxisome placed into cytoplasm. Each cell contains 100 or more than peroxisome present in both plant and animal. Contains more than 50 enzymes. The function of peroxisome first is hydrogen peroxide metabolism. These enzymes present in the peroxisome both lead to the production and elimination of H2O, which is reactive oxygen species. Second is fatty acid oxidation. Oxidation of fatty acid in animal cells occur in both peroxisome and mitochondria, but in yeast and plants only limited to peroxisome. Lipid biosynthesis. Synthesis of cholesterols and dolichs occur in both ER and peroxisome. Bile acid synthesis takes place from cholesterol in the liver. 4 is germination of seeds. Peroxisomes in seeds responsible for the conversion of stored fatty acids to carbohydrates, critical to providing energy and raw materials for the growth and of germinating plants. 5. Photorespiration. Peroxisomes in leaves, particularly in the green ones, carry out the photorespiration process along with chloroplast. 6 is degradation of purines. Carry out the catabolism of purines, polyamines, and amino acids, especially by uric acid oxidase. 7. Bioluminescence. Luciferase enzyme found in the peroxisome of fireflies help in bioluminescence, and those aid the flies in finding a mate or its meal. Next, let's proceed to peroxisome biogenesis. Peroxisome biogenesis encompasses the process of peroxisome membrane formation, importance of matrix proteins, and proliferation and inheritance of organelle, according to Exert and Erdmann 2003. And also, proteins responsible for peroxisome biogenesis have been designated peroxins, with PEX representing the gene's acronym, Distel et al. 1996. Here, Arabidopsis peroxisens are represented as at PEX to avoid confusion with yeast and mammalian
it was
peroxins.
thought that the membrane shell of the peroxisome is formed by budding of the endoplasmic reticulum, or ER, while the content or matrix is imported from the cytosol or cytoplasmic matrix. However, this is how now evidence suggesting that the new peroxisome always arise from the pre-existing ones, being formed by growth and fission of old organelles similar to the mitochondria and chloroplast. Those peroxisomes are a collection of organelles with a constant membrane and a variable enzymatic content. All of their proteins, both structural and enzymatic, are encoded by nuclear genes, are synthesized in the cytosol or cytoplasmic matrix. Examples on the free ribosomes. The protein present in either lumen or membrane of the peroxisomes are taken up post-translationally from the cytosol cytoplasmic matrix.
The glysosome are specialized peroxisome. Their main function is to convert fatty acid to carbohydrate. They are present in plant and fungi. They are prevalent in the germinating seeds in their fast-soaring
and oily rich
tissues.
seeds of many higher plants. They resemble with peroxisomes and morphological details, except that their crystalloid core consists of dense rods of 6.0 micrometer diameter. They have enzymes for fatty acids metabolism and gluconeogenesis, that is, conversions of stored lipid molecules of spirosomes of germinating seeds into the molecules of carbohydrates. Functions of glyoxysomes perform following biochemical activities of plant cells: fatty acid metabolism and glyoxylate cycle. Fatty acid metabolism: during germination of oily seeds, the stored lipid molecules of spirosomes are hydrolyzed by the enzyme lipase glycerolester hydrolase to glycerol and fatty acids. Glyoxylate cycle: the glyoxylate pathways occur in glycosome, and it involves some of the reactions of the Krebs cycle, in which citrate is formed from oxaloacetate and acetyl-CoA under the action of citrate synthesis enzyme. The citrate is subsequently converted into isocitrate by a kinase enzyme. The cycle then involved the enzymatic conversion of isocitrate to glyoxylate and succinate by isocitratase enzyme.